Hi guys, in this video we're going to talk about the concept called protein hydrolysis. Now you may recall, in order to synthesize a peptide bond, you take two amino acids, bring the amino group, carboxylic acid, together. It's going to form an amide bond. However, if you want to reverse that process, so let's say we had, we're just going to kind of zoom in on a portion or a region of a peptide. Okay, so I'm just indicating that I'm not worried about what the rest of this is, but here we have our amide bond that's now connecting two different amino acids. And in prior videos we talked about how amides are quite stable. They have half-lives on the order of close to 100 years in pure water. So proteins are really quite stable. This is one of the reasons why nature has chosen the amide bond or the peptide bond as sort of the molecular glue to hold together our proteins. But if we wanted to break this bond, okay, and give back either peptide fragments or possibly chop the whole thing down to individual amino acids, there are several ways you could do this. Number one, you could use H+ and water. Okay, and typically you have to heat this. And usually the acid that's used has to be a very strong acid, like HCl or sulfuric acid. And you often have to heat this for 6 hours, maybe even overnight. And what this will do, it will hydrolyze to individual amino acids. Okay, so basically if you had a protein, let's say, that was 300 amino acids and its total primary structure, if you were to hydrolyze that using acid-catalyzed hydrolysis, you would then get 300 individual amino acids. So you can certainly do that with strong acid and heat. You can also do this using base-catalyzed hydrolysis, so strong base, H2O, and also heating this. And so when I say strong base, I'm talking about like 6 to 10 molar sodium hydroxide or potassium hydroxide, and then heating this overnight. Again, it would chop that 300 amino acid peptide down into 300 individual amino acids. So that's how you might do this in a laboratory. If you had a protein, you could treat it with either acid or base, and then get your collection of the amino acids that are in that protein. However, in our bodies and in many other circumstances, you don't necessarily need or want to have to treat this with such caustic reagents. And so oftentimes what's used to degrade a protein are enzymes. Okay, and the specific types of enzymes that you can use to break apart peptide bonds are called proteases. Okay, and there are a whole host of different proteases that will have different amino acid or sequence specificities that would allow you to break down specific bonds. But if you were to treat a peptide like this with a variety of different proteases, you would get individual amino acids and peptide fragments. Okay, because if you had a protein of say 300 amino acids, proteases again often have certain specificity. They're only going to cut say after or before a certain amino acid. So you might not be able to chop the entire thing down. But basically all of these processes are doing essentially the same thing. They're hydrolyzing a protein with water. So if I simply draw a water molecule up here, what's in essence happening is that water is reacting at the carbonyl carbon, and it's then breaking apart this amide bond to then separate the two components. So those are the main ways that you can undergo peptide hydrolysis. Fortunately for us, we don't have, in our bodies, we don't really have concentrated acid or concentrated base, except really in one location, and that's in our stomach. So our stomach has a pH around 1.5. So in your stomach, what's actually going on when you ingest proteins in food, this reaction right here is happening. The proteins are being hydrolyzed under acidic conditions. But in most other cellular environments, we have more of a pH around 7.4, more close to neutral water, And proteins are really quite stable under those conditions. All right, so that covers protein hydrolysis. Good luck.